Life is a mystery. It is an intricate web of interactions and processes that have evolved over billions of years. From the tiniest microorganisms to the largest mammals, life manifests in countless forms, each uniquely adapted to its environment. It is complex and diverse. The sheer variety of life forms on Earth is staggering, each playing a role in the delicate balance of ecosystems. Yet, despite this diversity, there is a common thread that unites all living things. But at its core, all life shares something fundamental: DNA. This remarkable molecule is the foundation upon which the diversity of life is built. It is the universal language of biology, encoding the instructions necessary for life. DNA is the blueprint of life. It is a long molecule made up of nucleotides which are arranged in a specific sequence to form genes. These genes are the instructions for building and maintaining an organism. It holds the instructions for building and maintaining an organism. Every cell in an organism contains a complete set of DNA, which guides its development, function, and reproduction. These instructions are passed down from generation to generation. Through the process of reproduction, DNA is copied and transmitted to offspring, ensuring the continuity of life. Think of DNA as a vast library. This library contains all the information needed to create a living being. Each book in this library is a gene, and each gene contains a specific set of instructions. This library contains all the information needed to create a living being. The information is stored in the form of genes, which are segments of DNA that code for proteins. The information is stored in the form of genes. These genes are like individual books within the library, each containing a unique set of instructions. Genes are like individual books within the library. Each gene carries a specific set of instructions that determine various traits and characteristics of an organism. Each gene carries a specific set of instructions. These instructions determine traits. Traits are the physical and biochemical characteristics that make each organism unique. Traits are things like eye color, height, and even susceptibility to certain diseases. These traits are the result of the expression of genes, which interact with the environment to shape an organism. Understanding DNA is crucial for understanding life itself. By studying DNA, scientists can uncover the secrets of how life works, how it evolves, and how it can be manipulated for the benefit of humanity. The story of how we came to understand DNA is a fascinating one. It is a tale of curiosity, perseverance, and groundbreaking discoveries that have revolutionized biology and medicine. It's a story of scientific breakthroughs, collaborations, and rivalries. From the discovery of the double helix structure to the sequencing of the human genome, the journey to understand DNA has been one of the most remarkable scientific endeavors in history. The middle of the 20th century was a time of great scientific discovery. Scientists were racing to unlock the secrets of life. One of the biggest mysteries was the structure of DNA. We knew DNA carried genetic information, but how was this information structured and passed on? In the 1950s, several scientists were working on this puzzle. Among them were Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick. These four scientists would ultimately play a pivotal role in one of the greatest scientific discoveries of all time: the double helix structure of DNA. Their journey was not without its challenges. It was marked by competition, collaboration, and controversy. But ultimately, their work would change the world forever. Rosalind Franklin was a British chemist. She was a pioneer in X-ray crystallography. This technique uses X-rays to study the structure of molecules. Franklin's work was crucial to the discovery of the DNA structure. Franklin created high-resolution X-ray images of DNA. These images, particularly photo 51, were instrumental. They revealed the helical structure of DNA. Franklin's work was meticulous and groundbreaking. Unfortunately, Franklin's contributions were not fully recognized during her lifetime. She faced gender discrimination in the male-dominated field of science. However, her work laid the foundation for one of the most important scientific discoveries in history. Maurice Wilkins was a New Zealand-born British physicist. He worked closely with Rosalind Franklin at King's College London. Wilkins was also interested in DNA structure. He played a critical role in facilitating the discovery. Wilkins shared Franklin's X-ray images with James Watson. This act, though controversial, proved to be a turning point. The images provided Watson and Crick with crucial information about the DNA structure. Wilkins, along with Watson and Crick, shared the Nobel Prize in Physiology or Medicine in 1962. This was in recognition of their work on DNA. Section 5. Watson and Crick, a partnership for the ages. James Watson, an American biologist, and Francis Crick, a British physicist, were working at the University of Cambridge. They were determined to solve the DNA puzzle. Watson and Crick used a model-building approach. They relied heavily on the work of Franklin and Wilkins. Their partnership was one of intellectual synergy. Watson brought a deep understanding of genetics. Crick contributed his expertise in X-ray diffraction patterns. Together they were able to synthesize the available data. They proposed the now famous double helix model of DNA. Their discovery, published in 1953, revolutionized biology. It opened up entirely new fields of study. These fields include molecular biology and genetics. Section 6. Unraveling the helix, a scientific revolution. Watson and Crick's double helix model was simple yet elegant. It explained how DNA could store genetic information. It also showed how this information could be replicated and passed down through generations. The double helix is often compared to a twisted ladder. The sides of the ladder are made up of alternating sugar and phosphate molecules. The rungs of the ladder are composed of pairs of nitrogenous bases. There are 4 types of nitrogenous bases in DNA: adenine, thymine, guanine, and cytosine. Adenine always pairs with thymine, and guanine always pairs with cytosine. This complementary base pairing ensures that the genetic information is copied accurately during DNA replication. Section 7. DNA, the building blocks of life. The discovery of the double helix structure of DNA revolutionized biology. It provided a fundamental understanding of how genetic information is stored, replicated, and transmitted. DNA is a polymer, a large molecule made up of repeating units called nucleotides. Each nucleotide consists of a sugar molecule, a phosphate group, and one of the 4 nitrogenous bases: adenine, thymine, guanine, or cytosine. The sequence of these bases determines the genetic code. This code provides the instructions for building and maintaining an organism. It's like a language that cells understand. Section 8. The legacy of the double helix, a new era in science. The discovery of the double helix structure of DNA by James Watson and Francis Crick in 1953 marked a pivotal moment in the history of science. This groundbreaking revelation has had far-reaching implications, transforming our understanding of genetics and molecular biology. The discovery of the double helix structure of DNA has had a profound impact on science and medicine. It provided the foundation for modern genetic research, enabling scientists to decode the genetic instructions that govern the development and functioning of all living organisms. It has led to countless breakthroughs, including genetic engineering. This field has revolutionized the way we approach biological research and medical treatments, allowing for precise manipulation of genetic material to achieve desired outcomes. Scientists can now manipulate DNA to create genetically modified organisms, or GMOs. These organisms have been engineered to possess specific traits, such as resistance to pests, diseases, or environmental conditions, which can significantly enhance agricultural productivity and sustainability. GMOs are used in agriculture to produce crops that are resistant to pests and herbicides. This not only helps in reducing the reliance on chemical pesticides, but also contributes to higher crop yields and food security. DNA fingerprinting is another breakthrough. This technique, developed in the 1980s, has become an invaluable tool in forensic science, enabling the identification of individuals based on their unique genetic makeup. This technique is used in forensic science to identify individuals based on their unique DNA profile. It has revolutionized criminal investigations, providing a powerful method for solving crimes and exonerating the innocent. It's also used in paternity testing and ancestry research. These applications have made it possible to establish familial relationships and trace genetic lineages with a high degree of accuracy. Gene therapy is an experimental technique that involves altering genes to treat or prevent disease. This cutting-edge approach holds the potential to cure genetic disorders by correcting defective genes or introducing new ones. It holds great promise for treating genetic disorders and other diseases. As research progresses, gene therapy could become a standard treatment for a wide range of conditions, offering hope to millions of patients worldwide. The discovery of the double helix structure of DNA has ushered in a new era of scientific understanding. It has opened up new avenues of research and innovation, leading to advancements that were once thought to be the stuff of science fiction. It has given us unprecedented insights into the nature of life itself. By unraveling the mysteries of DNA, scientists continue to push the boundaries of what is possible, paving the way for a future where genetic knowledge can be harnessed to improve human health and well-being. Section 9. Conclusion: the enduring power of discovery. The story of the discovery of the double helix structure of DNA is a testament to the power of human curiosity and ingenuity. It was a groundbreaking moment that forever changed the landscape of biological sciences. It's a story about the collaborative nature of science. Watson and Crick's work was built on the contributions of many other scientists, including Rosalind Franklin and Maurice Wilkins, whose efforts were crucial to the final discovery, and it highlights the importance of perseverance in the face of challenges. The journey to uncover the structure of DNA was fraught with obstacles, yet these scientists pushed forward, driven by their passion and determination. The legacy of Watson, Crick, Franklin, and Wilkins lives on. Their work not only earned them prestigious accolades, including the Nobel Prize, but also paved the way for future generations of scientists. Their discovery continues to shape our understanding of life. The double helix model has become a fundamental concept in genetics, influencing countless research projects and medical advancements. It fuels new discoveries and innovations that are changing the world. From genetic engineering to personalized medicine, the impact of understanding DNA is vast and transformative. As we continue to explore the mysteries of DNA, we can only marvel at the elegance and complexity of this remarkable molecule. Each twist and turn of the double helix holds secrets that could unlock new frontiers in science and medicine. The double helix is a reminder that the greatest discoveries often lie hidden in plain sight, waiting for a curious mind to unlock their secrets. It stands as a symbol of the enduring power of discovery, inspiring future generations to look closer, think deeper, and never stop questioning.